Thank you. Thank you so much. And hello everyone. I'm a Digital 30th student at the Department of Engineering Science, University of Oxford, and the topic for my presentation is viscoelastic assessment of porcine aorta across two length scales. So, aorta is the largest artery in the body that is supplying blood from heart to different organs, and these are basically the points about the structure of the aorta. So we have the outer layer of the aorta, which is known as tunica externa, and externa is mainly composed of collagen and is supported by these external elastic laminae. Then we have in between tunica media, which is made up of these smooth muscle cells, elastic tissue as well as the collagen. The characteristic of tunica media is that we have these muscle tissues which are arranged in an anisotropic manner, which implies that the circumferential properties are different as compared to the longitudinal properties. And then finally we have intima, which is supported by the internal elastic laminae, and we also have the endothelial cells in contact with this particular layer. So the aims and objectives of this particular research being done is to measure the viscoelastic properties of aorta at a macro and micro scale. This will enable us to better understand the aortic degradation, which is often associated with the chronic artery disease. And that will also enable us in understanding the results from elastography techniques, which is a non-invasive technique for measurement of the shear modulus and the elastic modulus. The technique being used for heart and aorta is known as dynamic microelastography, wherein we have the external acoustic transducers and we try to map the displacement of the smooth muscle cells. Then the other aim of this particular work is to develop these fractional order derivatives, which basically are very different from the traditional viscoelastic models. And we try to fit the response of the aorta in the frequency domain using these particular fractional order derivative models. In terms of application, this can have an application in the finite element analysis of the aorta when it comes to the pulsatile blood flow, because there often we use these viscoelastic models to compute parameters like the wall shear stress and the pulse wave velocity. And again, like I've mentioned, that the alterations in the structure and mechanical properties, once we understand them, we can also understand that how is it actually having an impact on the chronic artery disease. So this particular work is divided into two parts. One is the rheology work, the other one is the nano indentation work. Rheology work is pretty much at the macro scale, and nano indentation work is pretty much the micro scale. So in rheology, we did two types of experiments. One was the strain sweep experiment, the other one is the frequency sweep experiment. In the strain sweep experiment, we took three porcine aortas. We took like three big, sorry for writing here. It should be like porcine aorta. And the thickness of these samples was approximately 1.5 millimeters in average. We took an overall of 9 samples, and the location of these particular samples was from the thoracic region. Then we are immersing these samples in TVS at 4 degrees, and until the time we are performing these particular strain sweep test. And then finally we have these particular samples extruded using an 8 mm biopsy punch, as you can see on the right hand side. And you can also see the whole structure of the aorta there. We have the thoracic region, we have the abdominal region, and you can also see lungs there. And then what we do is we try to attach these particular samples to the rheometer, to the top plate of the rheometer. And in the bottom plate we have this metallic chamber in which we are filling the PBS solution, phosphate buffered saline solution, which is at room temperature. So the results are as follows. What we try to do from here is that we try to vary the shear strain from 0.01% strain until 10% until 4.2% strain, and we try to see that at what point of the strain are we getting to being in the viscoelastic region. So what we noticed in this particular case was that in these three bits we have, like if you take the average of these three bits, you can see that from between 0.01% strain to 0.1% strain, you will have a linear viscoelastic region, followed by a non-linear viscoelastic region. Now, that particular non-linearity can either be because of slippage, or it can potentially be because of the damage as well. And in the table, you can see that I have these particular values of different depths. So you can see that the storage modulus at 0.01% strain varies from 6 to 9 kilopascals. As we are increasing the strain to 4.2%, this particular value of the storage modulus decreases to 2.7 to 5.2 kilopascals. Loss modulus is fairly constant, like it's approximately 1.7 in tick 1, 1.5 in tick 2 as we are actually varying the shear strain. So once we know that we have the threshold value of this particular shear strain, We can also perform these frequency sweep experiments and say that once we are performing these frequency sweep experiments, our samples are pretty much in the linear viscoelastic region. So again, we put frequency sweep tests, and in this case, we put again 9 samples from thoracic region. And on the right hand side, you can actually see this particular equation of the spring pod. So spring pods are the fractional order time derivatives of the springs in dash pods. And equation 1 here is representing the stress-strain relationship. So you can see that it's a fractional order stress-strain relationship. In this particular equation 1, you can see the first term, that is e down to the power of alpha, that's known as the coefficient of consistence. And that is taking into account the rate of binding and the unbinding of these muscle tissues. Once we apply the Fourier transform to this equation 1, we can actually get the stress and strain in the frequency domain. And then we can sort of break down this particular complex modulus into 2 components. One is the storage component, the other one is a loss component. The storage component is given by equation 4, and we have a loss component which is given by equation 5. As you can see that through this equation, the characteristic is that both the storage and loss modulus, they are having a power law dependency on the frequency, which is why we can use this particular model to represent the behavior of aorta in the frequency domain. Again, as you can see, as far as the results are concerned, we have the storage modulus increasing from 9 to 5 kilopascals to 14 kilopascals in big 1, 5 to 8 in big 2, and approximately 6 to 7 in big 3. Loss modulus is fairly constant. There's not a strong dependency of loss modulus as far as frequency is concerned. So this is the overall graph when it comes to, like, I've taken the average values of the storage and loss modulus here and plotted that from this particular, like fitted that using this particular equation on the right hand side, which is representing the complex modulus in terms of storage component and the loss component. Now once we fit these particular models, we can also compute the parameters. Here alpha is known as a coefficient, like I mentioned before, coefficient of consistence, and alpha is a dependency of this particular modulus on the frequency. So you can see that this is like varying from 7.4 to approximately 14 kilopascals second to the power of alpha, and that is because there's a fair amount of variability when it comes to testing tissues from one pit to another. So once we perform the bacteriological experiments, we also were very interested in understanding the properties of the microscale, and that was the reason as to why a nano-indentation test was performed in Liverpool. And this is the schematic diagram of a nano-indentation setup, and on the right hand side you can see this particular G200 nano-indenter which is being employed here. And the concept is very simple. It's just that you apply a magnetic flux. Because of magnetic flux application, you have the indenter moving in the vertical direction. You have these capacitor gauges which are trying to measure the displacement of this particular indenter. And depending on the input frequency, you can then get the value of the storage and the loss modulus. So in this particular series, we had a 100 micrometer flat cylindrical punch tip, and all these tests were conducted at a frequency of 10 Hz. We had an oscillation amplitude, which is the amplitude of oscillation of this indenter, set to be as 0.5 micrometer, and the pre-compression was 5 micrometer in all these tissues. We did a total of 62 indentations, in which 22 were in the ascending region, 10 in the proximal descending, 10 in the mid descending, and 20 in the distal descending. And then this is the whole procedure in which we are trying to extract the rectangular samples, place them in the rig with a PVS solution, and then selecting the indent points and then finally performing the nano-indentation. So the results from these particular nano-indentation experiments are as follows: that we can see that in the ascending region, the storage modulus is 40 kilopascal. It reduces to almost 20 kilopascal in the mid-descending region, and then again increases in the distal descending region. As far as loss modulus is concerned, the loss modulus varies from approximately 4 to 7 kilopascals from the mid-descending to the ascending region. And damping ratio, it actually increased as we are going from the ascending region to the proximal descending to mid-descending and finally to the distal descending region. So damping ratio is the ratio of the loss modulus to the storage modulus, which takes into account the amount of energy that has been lost to the sample. So finally, the results, like the overall conclusions, are as follows: that we have a linear viscoelastic region until 0.1% strain as far as the macro samples are concerned on the macro level, and we saw that there was a monotonic increase in the storage modulus as a function of frequency from the rheological experiments. From the rheological experiments, we again observed that the loss modulus is independent of frequency, and then we use this particular spring pod model, which has a very high correlation coefficient when fitting the frequency domain data. The correlation coefficient was better than 0.99. Then we had this particular fractional order, which varied from one bit to another by almost by a factor of 2, from 0.07 to 0.15. And then the interesting part is that at the micro level, we have the shear modulus, which is basically higher as compared to the macro level. So that's because potentially I feel that it's the collagen which is playing more of a role at the micro level as compared to that at the macro level, where we have a combined effect of the springs and dashboards. And then finally, I would like to thank supervisor of this particular project, Dr. Riaz Akhtar, Martin, who did these experiments, some experiments from nanoimitation, Professor Robin Cleveland from Department of Engineering Science, who is my main PhD supervisor, and NIH for funding this research. Thank you.